Hi everyone, in this video I am going to explain you about root anatomy, transfer section of monocotyledonous plant root. The internal structure of a typical monocotyledonous plant root. The internal structure of a typical monocotyledonous root shows following features. The very first one, epidermis. Epidermis is uniseriate or single layered and it is outermost layer. It comprising tubular living components. Any root, either it is monocotyledonous or dicotyledonous, cuticle and stomata are absent. Unicellular root hairs are formed due to elongation of some cells of epidermis. The root hairs of root are unicellular, thin, delicate structures which helps in the absorption of water and minerals from soil. Here you have to remember epidermis is single layered, outermost layer, cuticle and stomata are absent. The second one, cortex. The cortex of monocotyledonous root is made up of parenchymal cells with intercellular spaces. Checkpoints. The cells of outer part of cortex are soberanized in old roots. Soberon is the substance which is impervious to water, impermeable to water. It is called exodermis. Exodermis is found in most of the monocotyledonous roots. Cortex is made up of parenchymaceous cells with intercellular spaces. The outermost layers of cortex is called exodermis. Exodermis cells are soberonized cells which are impervious to water. Exodermis mostly found in monocotyledonous plant roots. Number 3, endodermis. The innermost layer of cortex is known as endodermis. Casparian strips are present in radial and tangential walls of endodermis. These strips are made up of soberin and called Casparian strips. These Casparian strips are Casparian bands first discovered by or first observed by the scientist Caspari. The cells of endodermis which are present in front of protoxylem lack of Casparian strips. These cells are called passage cells. The remaining cells are soberonized cells which are impervious to water, but passage cells lack of soberin, so these are called passage cells. During water conduction, water transportation, water moves through these passage cells. Why? Because the remaining soberonized cells are impervious to water. The number of passage cells is equivalent to the protoxylem cells and the number of rows of root hairs equivalent to protoxylem cells. Passage cells provides path to absorbed water from cortex to pericycle. Checkpoints. Root hairs are linearly arranged on root apex. Casparian bands and passage cells are well developed in monocotyledonous roots. Endodermis acts as a watertight jacket and prevents radial conduction of water. These are very important points you have to remember. Fourth one, pericycle. It is few thick layered. It is composed of prosenchyma. Only lateral roots are originated from the part of pericycle which is lying opposite to protoxylem. Thus, lateral roots are endogenous in origin. These divisions are followed by anticlinical divisions forming primordium which grows to form a lateral root. Checkpoint. Adventitious roots are also endogenous in origin because these are originated from stellar region. The pericycle of monocotyledonous roots only helps in the formation of lateral roots. Fifth one, vascular bundles. Vascular bundles are radial, which means xylem and phloem situated in different radius, and exarch. Exarch in the sense protoxylem present towards periphery, metaxylem present towards center. These xylem and phloem are separate and equal in number, arranged alternatively, which means xylem, phloem, then phloem, like that manner. Number of xylem bundles are more than 6 in monocotyledonous plants, which is called polyarch condition. Exception: onion is monocotyledonous plant, but it contains 2 to 4 vascular bundles. Conjunctive tissue is absent in monocotyledonous roots. Vascular cambium also will not form in monocot roots because monocot roots does not show secondary growth. The sixth one, pith or medulla. In monocot root, pith or medulla is well developed. Here you have to remember about number of vascular bundles. Generally in monocotyledonous plant roots, the number of vascular bundles are more than 6, which is called polyarch condition. Conjunctive tissue is absent, there is no formation of vascular cambium. Coming to medulla or pith, in monocotyledonous roots, pith is well developed. Let's see the diagrammatic representation of the transfer section of monocotyledonous plant root. See, the outermost layer is epidermis. Some cells of epidermis produces unicellular, thin, delicate thread-like outgrowths called root hairs. Below to that, exodermis. Below to that, general cortex. Cortical cells are parenchymus with intercellular spaces. The innermost layer of cortex is called endodermis, which shows Casparian strips or Casparian bands. In endodermis, the cells which are non-suberonized, those are called passage cells. Below to endodermis, pericycle. Pericycle helps in the formation of lateral roots. Then coming to vascular bundles are more than 6, xylem and phloem arranged alternatively, and they situated radial manner, which means xylem and phloem situated in different radius. Protoxylem present towards periphery, metaxylem present towards center. This condition is called exarch condition. Then pith or medulla, monocotyledonous plant root shows well developed pith or medulla in the center of stele. The second diagram is outline or ground plan of the transfer section of monocotyledonous root. Let's check this diagram carefully for your better understanding. Here we have to see the differences between dicot root and monocot root. Most of the features are similar in dicot root and monocot root. First one, pericycle. In dicotyledonous root, pericycle helps in the formation of lateral roots and also helps in the formation of lateral meristem, which leads to helps in secondary growth of root. But in case of monocot root, pericycle only helps in the formation of lateral roots. There is no secondary growth in monocotyledonous plants. Second one, vascular bundles. In dicots, the condition of vascular bundles is diarch to exarch. But in case of monocot root, polyarch. Third one, cambium. In dicot root, cambium develops at the time of secondary growth, but there is no formation of cambium in monocot root. Why? Because there is no secondary growth. Coming to pith. Pith is also called medulla. In dicot root, pith is absent or poorly developed, which means inconspicuous. But in case of monocot root, pith or medulla is well developed or fully developed. Up to here, root anatomy. Under this, transfer section of dicot root, transfer section of monocot root, both were completed. In next video, I will explain you about stem anatomy, transfer section of dicot stem, transfer section of monocot stem. Thank you very much.